Good afternoon, everyone. On behalf of Astrea Bioseparations, I would like to welcome you all to the webinar titled Downstream Process Intensification for Challenging Large Modalities in Cell and Gene Therapy Bioprocessing. I am Abishek, a conference producer at IMAPAC, and your host for the day. I would like to share a little bit about us. IMAPAC strives to be your one-stop shop for the biopharma industry's business intelligence and networking opportunities. Over the past 14 years, we have established ourselves firmly as a trusted organizer of high-quality biopharma B2B conferences and exhibitions in Asia Pacific and beyond. In the recent 2 years, we have exponentially expanded our portfolio to include digital marketing solutions, market research reports, and our webinar series. Today, we are hosting the Downstream Process Intensification for Challenging Large Modalities in Cell and Gene Therapy Bioprocessing webinar, powered by Astrea Bioseparations. Here's a quick introduction on Astrea Bioseparations. Astrea Bioseparations, also a biotech company, is a leading provider of chromatography solutions for the life sciences industry. With products used in the manufacture of over 20 approved therapeutic and medical products, Astrea Bio is trusted by scientists around the world to deliver precise and reliable results. Astrea Bio is headquartered in Cambridge, UK, with manufacturing facilities at the Isle of Man, British Isles, Joliet, Quebec, Massachusetts, USA. I would like to express my heartfelt gratitude to Astrea Bioseparations and our distinguished speakers for making this webinar possible. Thank you so much for your time and commitment to be a part of this webinar. And to all the attendees, thank you for taking the time out to be with us here today. Before we start, here are a few housekeeping announcements. We strongly encourage everyone to submit your questions by using the Q&A function at the bottom of your screen. The representatives from Astrea Bioseparations will be answering them during the live Q&A session after the presentations. So now, without further ado, I'd like to introduce our first speaker for the day, Dr. Marc Hummersone, Senior Director, Research and Development at Astrea Bioseparations. For introductions, Dr. Marc Hummersone has over 20 years of project and managerial experience in both big and small technology companies, including industry leaders like AstraZeneca, GE Healthcare, and Cytiva. He has a broad experience base across a wide range of technologies, including the development of novel chromatographic media for the purification of complex biological feed streams, as well as drug discovery and development. He was a key contributor to the development of the fibrochromatography family of products, which resulted in the acquisition of Purify by GE Healthcare in 2017. Additionally, he was a key part of the management team who validated the concept of synthetic lethality and who identified and developed Lynparza, which is an approved drug for the treatment of BRCA-mutated advanced ovarian cancer, which resulted in the acquisition of Kudos Pharmaceuticals in 2005 by AstraZeneca. Marc has held positions including Company Director, Chief Scientific Officer, and Department Director. Dr. Marc, the floor is now yours.
Well, thank you, Abishek, for the introduction. And I can now start my presentation. My name is Marc Hummersone. I'm the Senior Director of R&D of Astrea Bioseparations, and I'm here to talk to you today about the downstream process intensification for challenging cell and gene therapy bioprocessing. And there are increasing approvals of larger eligible patient populations within the cell and gene therapy space. But what are we doing about it? To put this in perspective, let's look at how approvals have evolved over the years. The first 8 US gene therapies for rare genetic diseases approved by the FDA have a combined estimated eligible patient population approaching 18,000, while in the EU, the estimated number approaches 16,000. Several gene therapies are in the pivotal stage of clinical pipeline and could receive a regulatory decision during 2024 to 2026. And if approved, they could thrust non-oncology gene therapy into the space of high demand and requirement from the point of view of the manufacturers. To give an example of this, please see the slide which shows the estimated patient population of the first 8 FDA-approved rare genetic diseases. And a great example of this is Lyfgenia, with a patient population of 20,000. However, compare that with some of the indications that are in late-stage clinical trials for non-oncology indications, things like diabetic retinopathy with a 20 million eligible patients, wet AMD with 5.7 million eligible patients, and diabetic peripheral neuropathy with 4 million eligible patients. The surge in demand means that we need to think differently about how we make the drugs to satisfy the market and, more importantly, patient demands. So what's currently stopping us from delivering the kind of scale that we discussed? There are challenges for cell and gene therapy manufacturing. These modalities are large compared to traditional small molecule therapeutics and even compared to monoclonal antibodies. These larger modalities are fragile too. The downstream processes, i.e. the purification of the molecule of interest from the biological feed stream that is required for their production, still uses technologies which were designed for other targets. And there is a lack of standardization across the industry, which sees producers that are all using different ways to make similar products. Their varied feed streams and issues are also varied for their purification. Examples of those yields that are deemed acceptable are shown for AAV and lentivirus, where we're accepting 50% and 25% as being acceptable yields, mainly because the purification step isn't efficient. And in many cases, it actually damages the products we're trying to purify. We must intensify the downstream purification process for cell and gene therapy targets in order to drive down cost and speed up delivery for patients who need these cutting-edge therapies at an affordable price. This is where I'd like to introduce you to Astrea Adept, which is a novel nanofiber-based chromatography media. It's been tailor-made for cell and gene therapy targets, just like the ones that I've described in the previous slides. It has a high binding capacity because it has a higher surface area, which allows us to apply more chemistry. It features very large pores, way bigger than resins, which allow high flow rates of media through them. And because of the open porous network, we have a low shear environment, which doesn't damage the delicate molecules that we're trying to purify. The pore size is carefully controlled as part of the manufacturing process. We have an average pore size of 860 nanometers, which allows lentivirus, adenovirus, exosomes, adeno-associated virus, and plasmid DNA, even up to large sequences, to freely permeate through the material. The material is manufactured according to a tightly controlled process to give us reproducible product time after time after time. I've told you about the Astrea Adept nanofiber material, but it doesn't stop there, because that nanofiber material has been developed into commercially available products for the production of plasmid DNA, lentivirus, and exosomes. These products are named pDNA Hero, LentiHero, and ExoHero, and they are available at a range of sizes and scales. It's these products that allow the user to efficiently purify otherwise difficult target molecules and also provide the ability to compress or intensify their purification process along the way. I told you about Astrea Adept nanofiber materials and how we've developed the Hero range of products that incorporate that nanofiber technology. I'd like to now show you how these products can help you, the user, at a range of scales. So let me start with plasmid DNA purification. Plasmid DNA is a critical component for cell and gene therapy. It's used for vaccine production, it's required for vector products, it's used as a linearized template for mRNA, and the markets are pretty big, where the CDMO market value is in excess of $250 million as of 2022. But the current trend is for larger and larger plasmid DNA sequences in order to carry more information in a single molecule. As such, we see an increase in viscosity of the feed stream as a function of the plasmid size, which often requires high operating pressures at the purification stage. And also, the larger sequences are easier to damage during the purification process. The plasmid DNA processing workflow is reasonably established and therefore more standardized than most other cell and gene therapy targets. A representation of a typical workflow is outlined here, where the upstream cell culture produces raw feed stream, and where the downstream processing part can be considered as an orthogonal process which features three elements: primary capture, a pseudo-affinity step, and a polishing step before the purified bulk plasmid DNA can be incorporated into the final product as required. So to give you an idea of the affinity of the pDNA hero, I'm showing you a smaller plasmid DNA sequence of 4.7 kilobases in size. The chromatograph shows when the unit line is switched to flow through the devices. We bind the plasmid DNA and the UV trace gradually increases as we start to achieve breakthrough. Now note the delta column pressure of the unit while we bind the target at a flow rate which is at 5 membrane volumes per minute, giving a residence time of 12 seconds. Now that's quick. Even at these flow rates, we have a dynamic binding capacity of 4 mg of plasmid DNA per mil of sorbent. Now I mentioned that the trend is for larger sequences. So let's consider a 34.7 kilobase sequence, but this time it's lysate, post-calcium fluoride treatment. If you look at the chromatography, you'll see that we load the lysate onto the nanofiber device at the same flow rate as before. That's a 12-second residence time. We load until breakthrough, then elute. Note that at no time does the delta column pressure rise above 0.15 megapascal. The elution peak is clean, as you can just about see from the gel, and the gel looks a little messy as the product in the elution peak is so big, and it's eluted with a 60% recovery. The take home from this slide is that we can take lysate of large plasmid DNA targets and purify by primary capture at speed and with good recovery each time, every time. Now, you remember that when I outlined the downstream process for plasmid DNA, there was an orthogonal element of that downstream purification process as part of the plasmid DNA workflow. Now, let's consider those workflows where primary capture using a resin DEAE is appropriate, followed by a polish with the pDNA hero product. In the initial primary capture step, you can see the Astrea pre-packed Purabead DEAE column. We're able to remove the RNA in the flow-through and then elute the plasmid DNA. And you'll see that we have open circular and supercoiled plasmid DNA variants in the elution pool. But then we take the elution pool and then we polish that elution pool with the plasmid DNA hero device. You'll see chromatography for 2 different scales of device, the 1x on the left and the 10x on the right. And note that the chromatography is identical, showing that the performance is completely scalable between size of device. With these devices run with a residence time of 12 seconds, we're able to remove the unwanted open circular plasmid variant to leave polished, pure, supercoiled plasmid DNA. And you'll see from the gel and the data table that the combination of IEX and fiber allow for the efficient and scalable purification of plasmid DNA in a highly purified manner. The data outlined in this slide allows us to quantify the efficiency of the process in terms of removal of impurities. The DEAE Purabead primary capture step shows the data for removal of DNA, endotoxin, HCP, and RNA, where there is significant reduction after the primary capture step, and further reduction is achieved with the polishing step. So in comparison to the lysate, we have virtually eliminated DNA, endotoxin, host cell protein, and RNA. I've shown you the performance capabilities of the Astrea Bioseparations materials and the pDNA hero range of products. This slide shows you the salt transition curves for the 1x, 10x, and 100x devices, where the chromatographic performance is virtually identical, regardless of scale. This is important as you scale your process and move towards manufacturing of drug substance, as it allows you to de-risk the scaling part of this downstream purification. But what does that scale look like? Here you can see the data for the 100x device, and here you can see the chromatogram for the primary capture of DNA to give an indication of binding capacity and performance. Note that we're able to bind a solution containing 0.05 5 grams per litre of DNA, running at a constant flow rate, and the pressure remains constant. And there's no pressure spike during elution. The elution peak is tight, and as you can see from the axis, which is measured in time, you can see how quickly we can process large amounts of product. And in this example, we elute at a concentration of approximately 3 grams per litre with a runtime of 30 minutes. So to summarise, for plasmid DNA purification, an orthogonal approach, and we're able to accurately position the sizes and nature of products for the efficient bind and elute and polish with a combination of pre-packed IEX products and plasmid DNA hero products. This is easy to scale and it's commercially available. So for plasmid DNA purification, we're overcoming the issues that are presented to us by the viscosity, the complexity of the downstream part of the process. We're able to move more towards a standardized process that fits with existing systems. As mentioned before, the current trend is for increased payloads, and lentivirus offers a larger cassette than AAV. For this reason, it's also a vector that's been approved since 2017, and it's also a vector of choice for CAR T therapy, which is a pretty decent sized market. But when making this stuff, we often suffer from low and variable yields, and there are few standard approaches for the scale-up or removal of HCPs and DNA from the lentiviral preparation. It's also expensive to make and is a significant proportion of the cost of CAR T therapies. If we consider how lentiviral vector preparation is generated from upstream processes where we undergo cell culture and transfection before harvesting and treatment with nuclease, then clarification and purification by ion exchange chromatography, which can be carried out by resin, membrane, or monolith. But now we're able to substitute those with the Astrea Adept LentiHero set of products before polishing purification prior to use. I want to focus on the use of the LentiHero range of products because they're the best, and they can be used for the purification of both suspension and adherent expression systems. Here I show you some chromatography data where a suspension-based preparation was loaded onto a LentiHero device. The column was run at 5 membrane volumes per minute, which allows for quick purification. The chromatography is identical for an adherent preparation. We get good recoveries of both lentiviral vector preparations, and it's quick. With elution in low salt conditions, and because of the open porous network, which offers a low shear environment, we get good recoveries. As you can see in the table below, with a 54% recovery across both expression models. There's a lot of data in this slide, but essentially what I'm showing you is that as well as LentiHero working across expression platforms, it produces the same results across different clarification protocols. We were routinely able to get approximately 60% recovery functional lentiviral vectors where HCPs are significantly reduced and extraneous double-stranded DNA removed too, all while running at high flow rates while achieving very low delta column pressures. So in summary, the Astrea Adept materials that have been featured into the hero ranges of product allow us to amplify the downstream processing efficiency. We have flexible solutions for a diverse range of modalities. And this allows us to provide serotype-independent technologies and adaptable workflows. This intensified productivity allows us to combine disruptive technologies with existing single-use infrastructures to empower you, the user, to purify for the first time larger modalities with tailor-made purification solutions. As you can guess, I'm very, very excited about this technology, and I know what it can do for you, the customer, and for the patient ultimately. So I've delivered as much information and excitement as I possibly can. I've finished my talk, and I would like to thank you so much for your time. Thank you very much indeed.
Thank you, Dr. Marc, for the very insightful presentation. I hope the audience have learned a lot from the valuable sharing lesson. Moving on, we have our final speaker for the day, Dr. Sujeong Yang. For introductions, Dr. Sujeong Yang is a subject matter expert in viral vector applications at Astrea Bioseparations. Sujeong plays an important role as an expert advisor in both upstream and downstream bioprocessing of viral vectors. With over a decade of dedicated research experience, Sujeong has been a key contributor to the development of gene therapies centered around viral vectors. Her journey began in the field of neuroscience at the University of Cambridge, where she earned her PhD in the subject. Following her doctoral studies, Sujeong continued her academic pursuits for gene therapies via viral vectors as a postdoctoral researcher and then as a fellow, all within the University of Cambridge in the UK. Dr. Sujeong, the floor is now yours.
Thank you for the introduction, Abishek. I'm Sujeong Yang, and I work as R&D scientist specializing in viral vector applications. In this presentation, I will be exploring two case studies focused on maximizing recovery with robust impurity clearance for lentiviral vector and exosome processing. Here, we introduce LentiHero, a new platform powered by Astrea nanofiber technology. On the left, you can see an SEM image of the nanofiber, which features a wide flow path of around 860 nanometer. This design allows for a high binding capacity of target molecules, a faster flow rate, and a low shear environment. LentiHero is an efficient platform for lentiviral purification, utilizing Astrea nanofiber with weak anion exchange chemistry. It has developed from the bench-scale spin column Nidius LentiHero to the larger radial FPLC devices LentiHero 1 and 10 for process development. The diagram on the right-hand side is the general workflow for LV purification. Following upstream processing, the conventional purification methods using an ion exchanger on existing platforms, such as resin, membrane, and monolith, can be replaced with Astrea nanofiber. This is the study outline from upstream to downstream process. We generated our own suspension LV feedstock, and here is a detailed overview of our upstream protocol. The batches undergo a series of clarification steps. Typically, the feed is first centrifuged, followed by 0.4 micrometer pass filtration, and then a buffer exchange through diafiltration. These clarified feeds were purified with LentiHero 1 capsule. Here, you can find the specifics of the buffer conditions used for FPLC. Unlike a strong anion exchanger, it only required low salt dilution and neutral pH. We used 0.6 molar sodium chloride in elution buffer. The samples were tested by analytics as listed here. To maximize the lentiviral recovery yield, we optimized the clarification methods prior to the primary capture by LentiHero. We studied to establish a set of centrifugation speed of the harvest and ultrafiltration molecular weight cutoff for diafiltration, which did not damage infectious LV particles prior to the chromatography primary capture step. In the workflow, you can see here, the lentiviral culture was centrifuged down. Only the supernatant went through pass filtration. Then we do buffer exchange using flat sheet or spin column-based ultrafiltration. Then the feed is purified with LentiHero 1. The table shows the conditions we tested. Then we measured infectious LV titer after these clarifications. As shown in the graph, condition 3 gave the highest TU compared to the harvest. Thus, this condition 3 was adopted for clarification workflow prior to LentiHero 1 chromatography. We also optimized the buffer exchange volume during diafiltration step using 100 kilodalton molecular weight cutoff spin column. We compared feeds that underwent buffer exchange volumes ranging from 1 time to 3 times. Then we measured the infectious LV titer TU, host cell proteins, and double-stranded DNA in the clarified feed. As shown in the graph here, using 1 time buffer volume resulted in the highest TU titer with more than 40% of impurities removal. When buffer exchange volume increases, the impurity level is lowered, but you also lose the infectious titer. Following these clarifications, the feed was purified with Lentihero 1 capsule at process development scale. Batch 1 and 2 were generated under the same transfection system. The table here shows 2 recommended loading capacities for Lentihero purification depending on the feed. You can find the loading and elution details in the table. We assessed the functional LBB recovery as shown in the graph. Both batches and loading conditions resulted in a consistent TU recovery of around 46%, with a TU yield reaching up to 8e�9 in total. We also noted that TU recovery can be influenced by biological batch variability and the clarification step. We focused on measuring impurity levels, and as shown here, Lentihero 1 consistently removed 98% of host cell proteins throughout the purification process. Additionally, the process achieved over 95% clearance of double-stranded DNA through pass filtration and buffer exchange via dye filtration. Overall, Lentihero purification effectively cleared DNA and host cell protein impurities, even with significant variations from the clarification step. This is our findings of Lentihero 1 purification at PD scale. First, robustness for infectious LBB recovery across the platforms such as cell lines, transfection systems, clarification. And the functional recovery yield was up to 8e�9 TU total. And average recovery was 46%. It has excellent impurity clearance. Host cell proteins removal was robustly higher than 98%. Double-stranded DNA removal was average 95%. Moreover, it maintained lower delta pressure during the repeated runs of chromatography. We extended the repeated cycles to 18 runs, and functionality measured by BSA binding capacity was not altered by multiple cycles of CIP. The next case study focuses on isolating exosomes using Astrea nanofiber technology. Extracellular vesicles, including exosomes, are becoming increasingly important in ATMPs because they offer versatile applications. They can be used directly in cell therapies or as vectors for delivering nucleic acids and other therapeutic agents. However, we still face challenges in achieving consistently high yields and purity at a large scale. This slide outlines the workflow for producing and isolating exosomes. We generated extracellular vesicles from 2 cell lines, human mesenchymal stem cells and HEC293 cells. The culture feedstock was clarified and diafiltered before being purified using Exohero nanofibers. The elution buffer contained 0.4 to 0.5 molar sodium chloride with a loading concentration of 2e⁻10 particles per mil. The analysis was carried out using the assays listed here. These studies were conducted in collaboration with our research partners. Current EV purification methods still face challenges. Bead-based adsorbents and size exclusion techniques are not practical for large-scale processes and struggle with resolving impurities. Commercial convective adsorbers are strong anion exchangers, so require high salt concentrations for eluting macromolecules, which often compromises EV stability. In contrast, ExoHero uses a nanofiber adsorbent with a weak anion exchange ligand, allowing low salt elution. This also allows pH adjustment of elution and binding buffers for greater selectivity. This graph shows EV isolation yields from mesenchymal stem cells and HEC293 cultures, ranging from 1.4 to 1.8 e⁻12 particles per mil of adsorbent achieved by ExoHero. This table summarizes the recovery and purity of EVs from two different sources, with data kindly provided by EcoBiotech and Evazome. As you can see, the results show a 50% recovery from mesenchymal stem cell feed and around 60% recovery from HEC293 feed achieved through two-step elution process. Purity analysis shows a significant improvement with a purity fold increase of over 2. These results suggest that ExoHero can effectively separate EVs from protein impurities with greater efficiency compared to current methods like size exclusion chromatography or strong anion exchanger. EVs can be separated based on their relative charge. This chromatogram shows the isolation of HEC293-derived EVs, a result generated by Emma Berman, a PhD student at UCL, sponsored by Astrea Bioseparations. You can clearly see two distinct elution peaks on a 100% gradient to 1 molar sodium chloride, highlighting the heterogeneity within the EV population. This enhanced resolution of EV subpopulations was achieved with ExoHero nanofibers. EMMA is currently continuing the characterization of these subpopulations of EV. And importantly, total recovery of EV in this experiment was 85% of the total load. Lastly, I want to emphasize that this technology will open a new chapter of efficiency and accessibility in bioprocessing complex feed streams. This novel nanofiber adsorbent enables fast and efficient purification of raw materials for advanced therapeutics. We offer 3 device ranges: LentiHero, ExoHero, and pDNA Hero. They are purifying lentiviral vectors, exosomes, and plasmid DNA, respectively. This technology can accelerate the discovery, process development, and manufacturing of target molecules. That concludes my talk. Thank you for listening.
Thank you, Dr. Sujeong, for the in-depth sharing on multiple case studies. Once again, attendees, if you do have any questions regarding the presentation, please submit your questions by using the Q&A function at the bottom of your screen. And now we'll proceed to the live Q&A session. We are glad to have Mr. Xiao Cheng, Field Application Specialist from Astrea Bioseparations, with us to answer your questions. So before we move on to the Q&A, here's a bit about Mr. Xiao Cheng. Xiao Cheng is a Field Application Specialist at Astrea Bioseparations with over 10 years working experience in biopharmaceuticals. His professional background helps the corporation provide full support in chromatographic solutions in APAC. He also holds his master's degree in microbiology from the University of Bonn, Germany. He has worked in several biotech companies, including Wuxi Biologics, Sartorius, Cytiva, and Oricel as a downstream process scientist. He has now devoted himself to purification technologies such as chromatography and filtration. All right, then. I think we have some questions coming in already. So let's start off with the Q&A session. Okay. So for the first question of the day, Mrs. Xiao Cheng, it's by an anonymous attendee, and the question reads, what are the unique properties of your Astrea Adapt nanofiber compared to other fiber membrane?
Hello, everyone. Yeah, thank you very much for attending the online seminar that hosted by IMAPAC and Astrea Bioseparations. And I'm Xiao, and exciting to have some idea exchange in the Q&A session. Yeah, and for the first question that is mentioned by Abby, so what is the feature for our nanofiber compared to other fibers or membranes you can find in the marketing? Yeah, that's very good questions. Generally speaking, you can expect very different pore size and materials from our nanofiber. We have this nanofiber with the chemicals in cellulose acetate, and during the manufacture, it combines two copolymers, and one polymer is responsible for the flexibility of these nanofibers. For example, it has very large pore size in 860 nanometer in average. And even so, another copolymer is responsible for the chemical and physical compatibility or strengths. And I will give you an example for that. For our nanofiber, you can use, for example, very high content of sodium acetate. For 1 mole sodium hydroxide, you can clean it, and for several cycles, and it can extend the lifetime. And in other words, if you know something about our devices, 1 milliliter and 10 milliliter, it was pleated, it can be pleated, and then can put into the capsid. And then for the capsule filter, it's very easy to enlarge it or to scale it up. And therefore, for the scale-up process, it must be easy compared to other nanofibers. So that's what I can give you some answers in the nanofiber compared to others.
All right. Thank you, Mr. Xiao Cheng. Moving on, we have...
next one.
Yeah. The question reads, for lentivirus purification, is that column available for commercial scale? And this is from Tapasa.
I would like to say nowadays we only have 2 devices in the marketing. It's 1 milliliter in column volume, and another one is 10 milliliter in column volume. It's possible for your process, and 1 milliliter you can use for the screening or process development, and 10 milliliter is GMP compliant, and you can use it for your batch preparation or something small batch production. And for even larger columns, you can expect in next year, and its production or development is ongoing. Thank you.
All right. Okay, so I think that answers your question, Tapasa. Moving on, we have the third question. So the third question reads, for large-scale processing, centrifugation I'll repeat, sorry. For large-scale processing, centrifugation as a clarification method isn't scalable. Have you explored other alternatives for the LentiHero workflow? What would you suggest?
Yeah, I would like to say that by centrifugation combined with filtration, and you can use it at a very small scale in your lab. For example, you can have 100 or 200 milliliter suspension or adherent cell cultivations or harvest. You can use centrifugation, but if you want to scale it up, you can consider other technologies such as depth filtration. You can choose some different metrics for depth filtration. For example, you can use some cellulose-based filters with or without filter aids, or you can also choose some TFF process. So I know, or I found in some papers that some researchers in the lab that they use the continuous production process for the harvest, and they use the microfilters for TFF, or and then it can also clarify your harvest pools before loading to the Lentiviral. Or even you can also find some other filters such as PP filters or some glass fiber filters for the pre-filtration, and then combined with other filtration process.
Okay, thank you, Doctor. Thank you, Mr. Cheng. So I'll move on to the next question. Again, another question from Tapasa. The question reads, what are challenges we could expect during scale of lentivirus using this chromatography?
I understand this question that you should expect some advantages by using this nanofiber in your chromatography. And yeah, I think that Dr. Marc and Dr. Sujeong has shared several cases which indicate that by using this Lentiviral nanofiber, you can expect very high binding capacity. And in our previous studies, and we load 1 plus 9 to 10 technical unit to 1 milliliter column volume of this nanofiber. It must be higher than the Q memory or traditional resins generally used. And another one is that you can expect very robust removal impurities, especially in HCP. And in these cases, you can find that over 90% of the HCP content in your harvest can be removed only using this one-step chromatography nanofiber. And another one is that you can expect very fast process by chromatography. When you use a traditional resin, it can last for several hours. But if you use this nanofiber, you can expect the retention time in several seconds and flow very fast. And then you can achieve one cycle for the purification of only 30 to 40 minutes. So that's very fast compared to the traditional chromatography method.
Okay, all right. Thank you, Mr. Cheng, for that answer. So I think, Tapasa, again, your question has been answered. So moving on to the next question, the question reads, in your workflow, you perform diafiltration for buffer exchange with the feed before Lentiviral chromatography. Is this step essential?
It's a very nice question. Yeah, from my side, that depends on your process design for downstream. Yeah, I would like to say it's not essential for Lentiviral. That's because the TFF or concentration or buffer exchange, sometimes you only consider the volume reduction for your loading mass. During using your traditional resins, the loading volume should not be larger. That's because the load volume or flow rate should be very slow. And that is limited for the loading volume. And you want to do that and to concentrate and use the TFF process. And for Lentiviral nanofiber, it's not limited for the flow rate. You can expect several seconds retention time, and flow is 5 to 10 column volume per minute, and that's very fast. And you can even load 100 or several hundred milliliter in 1 hour for the 1 milliliter Davis. And therefore, it's not essential to conduct the TFF process by using nanofiber. But if you consider that the TFF process can be used for the removal of some impurities, and you can do that, but we don't recommend to do that before the lentiviral loading. Thanks.
Okay, thank you, Dr. Cheng, for the very clear explanation. So moving on, we have one more question. It reads, does the lentihero system work for other pseudotypes of lentivirus?
Yeah, we can also expect other phytotypes, and sometimes for the lentivirus, and most of the and I think it's the third or fourth generation of the lentivirus in the marketing and used for the cell therapy, and they use sometimes most part of this project are using a VSVG phytotype, and you can also have different surface proteins or some different enveloped proteins in the surface of lentivirus, and it also works by using lentihero. And in the meeting or seminar that's shared by Dr. Marc, you will know that the lentihero is based on VKNI exchanger. That means it can also work for the other phytotopes.
All right.
Yeah.
Thank you for the explanation again, Dr. Cheng. I think most of the questions that we've received today have been answered, and I think most of the doubts are clear. So attendees, once again, if you do have any other questions, please feel free to drop it here. Rest assured, the questions will be answered anyways, even after the webinar. So I would like to thank Mr. Cheng for clarifying the doubts of our attendees for the day. As much as we would like to address all the other questions from the attendees, in the interest of time, I think we'll have to wrap up today's session. Not to worry, as I said, any unanswered questions will be submitted to our speakers, which will be answered offline and shared with all attendees via email. So if you do have questions separately for Dr. Marc or Dr. Sujeong, please feel free to post them on the Q&A box, and we'll answer it to you via a separate email after the webinar. So I think that wraps up the Q&A session and the webinar itself. So I hope you all enjoyed being a part of this webinar and found the presentation and discussions very insightful. IMAPAC would like to extend our heartfelt gratitude to Astrea Bioseparations for making this webinar possible, and a big thanks to the audience for being a part and making this webinar a success. Again, on behalf of Astrea Bioseparations, thank you, and looking forward to welcoming you again at the next webinar. Thank you, everyone, and have a good day.
Thank you.